Hello everyone. Neurons are specialized cells that receive, process, and transmit the information. This is the image of a neuron. They receive input primarily through their dendrites. This is a dendrite. You can see here. These inputs come from axon terminals. Through neurotransmitters, it travels and reaches to the dendrites. And in the case of sensory neurons, it is from the stimuli or environment. So when a dendrite is stimulated, a positive ions, specifically sodium ions, flow into the neuron that we discuss in detail, because inside of the cell is normally more negative compared to the outside. If enough sodium flows in, electronic potential is created here. A small change in the voltage that spreads through a short distance. So imagine through axon hillock. So if we proceed through this, imagine placing a small voltmeter near the axon hillock, the region where the cell body taps into axon. At rest, the voltage around minus 70 millimoles. After stimulus, you might see a small upward bump in the voltage as a positive charge is spread. If this bump reaches critical level threshold, which is around minus 55 millivolts, it triggers voltage-gated sodium channels at the axon hillock. This sets off an action potential. Once triggered, the action potential generates itself and travels through the axons, sending the signal towards the next cell. But there is a challenge. How do you make the signal travel efficiently along the axons, like those in human spinal cords or even longer animals like giraffe or dinosaurs? The solution is insulation. So how the faster it can travel, because we need faster response. The solution is insulation. Just like electrical wire coated with rubber or plastic to prevent current leakage, neurons have a biological insulator called a myelin sheath. So these blue are the myelin sheath here, produced by common cells in the peripheral nervous system or oligodendrocytes in the central nervous system. Myelin prevents ions from leaking out and allows electrical signal to travel faster with less energy loss. However, there is a problem. If entire axons are wrapped in the continuous myelin, the signal would weaken when it is traveled. By the time it reaches the end, it might not be strong enough to trigger the next neuron. Nature solved this by leaving small gaps in between the myelin sheath called nodes of Ranvier. At each nodes, there are many voltage-gated sodium and potassium channels. The signal spreads passively. The signal spreads passively under this myelin and reaches the node. So signal passes through this myelin sheet, reaches the node, and triggers another action potential. So then this action potential is traveled through this myelin sheet, then trigger another action potential at nodes of Ranvier. So it boosts the signal again. Then it spreads to the next node. The process repeats. This makes it look as if action potential is jumping from the node to node. In reality, there is nothing jumps. The signal is continuously spreading and being regenerated at each node, because it will pass through the myelin. It is regenerated in the nodes of Ranvier, then it's passed through the myelin sheath, then it again regenerated at nodes of Ranvier. But because it appears so, this process is called saltatory conduction. It is from the Latin word saltare, meaning leap. So, but so this is a sky cells which produce myelin sheath. These are nodes of Ranvier, and this is the synapse where neurotransmitters travel. So we can see that in this diagram. So in the saltatory conjunction, actually nothing jumps. At this point, let's pause and clear up some common myths. So the myelinated in the neuron, there are myelin sheaths which are separated by nodes of Ranvier, and these the ion channels are located in between these myelin sheaths. So where the influx of sodium comes. So first of all, see why it is called the saltatory conduction. Why it is called? Why it jumps? Why it seems like it jumps from the nodes of Ranvier to another node of Ranvier? The answer lies in how impulse travels and how myelin changes the rules of conduction. In unmyelinated axons, voltage-gated sodium channels, which are these, are distributed all along. This as these myelin sheaths are not present, it is represented all along the nerve and all along the neuron. But here, these are only present in the nodes of Ranvier because they have this myelin sheaths, these membranes. When action potential in myelinated neurons, when action potential starts at one point, uh, uh, so first discuss in non-myelinated nerves. So when action potential starts to one point, sodium channel open and depolarize the membrane. The depolarization spreads to adjacent region, targeting the next sodium channel to open. 2F continues step by step along the axons. It is the row. It is like row of row, and this process is continuous but slow because every small segment of the membrane has to undergo depolarization. Here depolarization occurs, then repolarization occurs, then depolarization, then repolarization. So this process is quite slow. Now in myelinated axons, the story changes. Myelin is a fatty insulating sheath wrapped around the axons of specialized cells. This job is to prevent the ions from leaking out the membrane. Voltage-gated sodium channels, as in myelinated nerves, are not present everywhere, but are concentrated at nodes of Ranvier. So what happen when action potential starts here? At one node, sodium channel open and depolarize the region. The positive current that flows inside the axon does not immediately fade away because myelin insulation prevents this leakage, which is not present in non-myelinated neurons. The This current travels rapidly beneath the myelin until it reaches the next node of Ranvier, and at the node the sodium channel open, regenerating the action potential. To observe, it looks like action potential skips from one node to another node. That's why it seems like jumping, but in reality, the local current flowing smoothly under the myelin and the signal is only reaches at nodes. So there are several factors these myelinated nerve fibers conduct rapid nerve impulse, which we discussed. Insulation is one of them because myelin sits very current, little current leaks out, so depolarized waves spread faster. And there are very few active zones in unmyelinated nerve fiber. Every bit of membrane must depolarize. In myelinated fibers, only the node of Ranviers need to fire, saving more time and energy. Electrical properties: myelin increases membrane resistance and decreases capacitance, allowing faster spread of electrical signals. So there is a rapid conduction, sometimes 100 meters per second in large myelinated axons. So does anything actually jump? The answer is no. Nothing physically leaps from one node to another. What happens is the action potential is then regenerated at nodes while current spreads invisibly under the myelin. The term saltatory from the Latin but Saltare describes how it looks, not what truly happens. Neurons are basically engineered from local electronic potentials, threshold firing at axon hillock to insulating power of this myelin and boosting effect of every feature ensures fast and efficient signal transmission. Saltatory conduction does not involve real jumping, just a smart design and let humans animals perform rapid movements, reflexes, and complex brain functions. Thank you.